Great. Thanks for the invitation. And I'll be talking about the intraoperative molecular diagnosis of brain tumors and beyond, and some other tumor types. So just to define the molecular, and we are going to talk about the copy number alteration and methylation profiling. And intraoperative is like a vague term, but right now if we are dealing with craniotomy within like 2 hours, time frame is a very reasonable time frame. So this is conflict of interest. So just to give you some background of copy number variation, it's actually a major structural variant that's very prevalent in the human genome, and it influences the gene dosage by the chromosomal gain, losses, or amplification. The size of the gene usually range from the 50 base pairs to a few mega base pairs. A lot of biological processes are involved, including metabolism and some of the neurological diseases. But most importantly, and probably most relevant to all the audience and me, is it's almost always universally detected in cancer genome. Now, multiple methods in clinic. Some of them are pretty comprehensive, like karyotyping, CGH, or array-based assay. Some are a little bit targeted, like the in situ hybridization or PCR-based methods. But they are either very time-consuming or very limited, and usually taking a couple days to weeks to get the result. So we asked, can we actually develop a fast and comprehensive high-resolution CNV assay? So if you think about the problem, very intuitively you say, okay, sure, let's just sequence the genome end-to-end. And obviously, many groups has done that using next-gen sequencing, PacBio, or even nanopore sequencing. The Stanford group a couple of years ago published this awesome nanopore sequencing, whole genome sequencing in 7 to 8 hours. But keep in mind, you need to load 48 flow cells at the same time in order to reach this. And the data they generated is actually very challenging to analyze. So can we make it faster and cheaper? So if you think about this question again, if you want to assess the whole genome quantitative changes like copy number variations, you don't actually need to sequence end to end. You can actually basically fragment the genome into small pieces and then randomly sample a small fraction of it and then align back into the reference genome to assess the quantitative changes, like the shallow sequencing. So here there are two considerations. First of all, those small fragments need to be big enough to uniquely map to the genome. Theoretically speaking, anything bigger than 16 will be okay. And the other thing is that we want to do real-time interpretation, so Nanopore is the way to go. But Nanopore, we know that we can sequence the short fragments, but we feel that we shouldn't waste the long resequencing capacity. So we think again, can we actually make those fragments together, making this kind of chimeric reads in order to increase the efficacy of the sequencing result. So this is how we develop this method in our lab. It's actually involved a cocktail of researching enzymes that has compatible overhang that's mixed together to fragment the entire genome. But at the same time, because those ends are compatible, you are able to stick them together into chimeric read in one single reaction. So here's using the CTAG overhang as the example. You are able to run the simple electrophoresis and you see this kind of reconstruction of those chimeric reads. So we call this method iSCORED, reversible sticking compatible overhang to reconstruct DNA. So so this is our cartoon. So basically we treat the genome with iSCORED, fragmented them, and then also reconstructed them, and then sequence with nanopore, analyzing to assess the quantitative and also the qualitative, the methylation data. So this is just an example using the characteristic 1p19q co-deletion in oligodendroglioma. You can see here that if you compare to the native DNA, you actually get more than 16x more fragments if you use the iSCORED. And this is just a plot of the chromosomal operations, and then you will see the 1p19q co-deletion. But if you sequence the native DNA, it's just much more variability. And we can actually get this done within 90 minutes. So yes, it works for detecting this kind of large chromosomal gain or losses, but we ask next, can we actually increase the resolution to those kind of smaller oncogenes? And if you think about this question, this is actually not simple because what I'm doing is basically mapping those small fragments into predefined bins. And the restriction enzyme is not really uniformly distributed across the genome. So if you do this, actually the baseline is not even and you get very huge variability. Coefficient of variation is about 0.68. It doesn't matter if you sequence more data, it just doesn't get lower than 0.6. So we ask, can we actually normalize to some of the predefined controlled genomic DNA? And we did that. And then by doing that, we are able to actually lower down the coefficient of variation to 0.1 to 0.15. At this level, we will be able to reliably assess the quantitative changes of those smaller amplifications. So encouraged by these results, we actually use we first use one NGS validated tumor with cyclin E1 amplification, and with the iSCORED sequencing, and basically we're able to detect this cyclin E1 amplification and also the exact region where it was amplified. We want to push this to the limit, so we do the titration experiments and to see how confident can we call this amplification with different tumor purity. So we did a lot of titration, and then bottom line is that using a z-score of 10, which is crazy specific, then we are able to get this consistent copy number alteration with 5% tumor content with 500K fragments. This is superior to the existing next-gen sequencing because most of them has a cutoff like 15 to 20% of tumor content to get of this kind of gene amplification. So we did a retrospective cohort of 26 brain tumors, get 100% concordance comparing to the oncoscan and also the next-gen sequencing results. Going back to the methylation, where's the methylation, right? So methylation, because methylation will get the methylation data at the same time, so we will be able to compare our methylation data to the existing atlas, and we compare a few reference, like using the Replixians or the Sturgeon. We found that Sturgeon is better than the other one, so we decide to use Sturgeon to actually help with our methylation classification. All right, so I'm going to give you a quick example of a brain tumor. This is a 73-year-old female presented with seizure, found to have a right temporal heterogeneous enhancing mass. Intraoperatively, we do this kind of morphology-based diagnosis. So a pathologist, a neuropathologist will tell you that this is a hypercellular glial neoplasm. That's it. Intraoperatively, we'll tell that this is a high-grade glioma. But if we can we did extract the tumor DNA and run our assay, and we get this characteristic 1+7 minus 10 alteration, and also the EGFR amplification in this sample, the methylation classification accurately classified to the glioblastoma with high collaborative score. And we wait 2 weeks, and then the oncoscan result came out pretty consistent with what we saw here. Another 1 week, next-gen sequencing result came out, and it has this EGFR amplification. But anyway, we already know that like 2 weeks before, and this is already a glioblastoma. So we implement this clinically to look at 15 challenging brain tumors that we encountered clinically. This is just the workflow and the time. And as you can see here, the morphology is usually pretty challenging, right? Usually called high-grade glioma or some other like glioma, just very, very vague terms. By using the methylation, we are able to call those specific tumors. And also the CNV actually help with defining those kind of characteristic CNV to help with the diagnosis. And the entire pipeline from the time we got a specimen to the time of report is about 105 minutes. So they can certainly be implemented clinically. And we are now doing that for other tumor types in Dartmouth, and all of the tumors that is frozen comes, and then if we have good enough tumors, we will do this copy number alteration. And then the goal is to give this information back to the clinical team within 2 hours to help with the diagnosis and some of those treatment options. So to conclude, we developed this iSCORED. This is a one-step DNA reconstruction method, and with nanopore sequencing, we are able to detect the CNV very easily, cheap, powerful. But when I say that, my colleague actually correct me, say, no, this is actually the so far the cheapest, fastest, and most powerful genome-wide CNV method. Okay, and concurrently we have the methylation to help with the classification in brain tumors. So what does it mean? We can actually tell the surgeon what that is and some of the targetable, possibly targetable mutations intraoperatively, so they can treat the patient even faster or even intraoperatively in the future. So other mutations like point mutation is compatible. Gene fusion is a bit challenging. Homologous deletion is a bit challenging. But lastly, I would like to share this with you that we actually put all of these bioinformatics pipeline, established this one. It's a HIPAA compliant cloud system. So if you guys are interested in implementing in your own institution, please talk to me. So finally, I would like to thank my graduate student, Francisco, and also the pathology resident, Aziz, for all of the bioinformatics works, and also collaborators and some supports. Thanks very much.